Hey everyone, and welcome. Today, we're diving deep into an organism you've probably seen on your benchtop, but maybe under a different name. Stutzerimonas stutzeri. Now, this entire explainer is powered by a pretty comprehensive literature search using Gemini Deep Search. And if you're the type who loves to go straight to the source, don't worry, the full notebook with all the references is linked for you.
Okay, so here's our game plan. We're going to start with its new identity, then walk through its clinical side, the real nitty-gritty of lab ID, tackle the big challenge of treatment, and then wrap up with how to keep it in check in the hospital.
So let's just set the stage here. We are talking about an organism that's popping up more and more as a pretty significant opportunistic pathogen, but its story has a very recent and really important twist. And that's the twist, right there. An old bug has a new name and, honestly, a whole new reputation to go with it. And trust me, this is way more than just a name change in the LIS. It's a huge shift in how we understand its role in human disease.
To really get this organism, you've got to understand where it came from. Its whole taxonomic journey is, I think, a fantastic example of how modern genomics is basically rewriting the textbooks we all learned from. Just look at this history. It has been all over the place. It started as a bacillus way back in 1895, then spent most of the modern era as a Pseudomonas. But the really big change, the one that matters to us now, happened in 2022. That's when whole genome sequencing gave us the proof we needed. It showed the Pseudomonas stutzeri was just too genetically different from the main Pseudomonas group to stay there, so it got its own genus, Stutzerimonas.
And the tech that really sealed the deal was average nucleotide identity, or ANI. So what this does basically is it compares how similar the genes are between two different bacteria. The magic number is 95%. If the similarity is less than that, they're likely different species. Well, in this case, the ANI was way below that threshold. This was the high-res, undeniable evidence needed to say, yep, this deserves a whole new genus.
Okay, so now that we know what to call it, let's talk clinicals. Who's actually at risk for a Stutzerimonas stutzeri infection, and what does it actually look like in a patient?
This thing is a classic opportunist. It can cause this huge range of pretty serious infections, but almost always in patients who are already vulnerable. Think post-op wounds, patients with lines and tubes, or anyone who's immunocompromised. It really seems to like bone and joints, but as you can see, it's a versatile pathogen that can cause trouble just about anywhere.
Now this is what I find really fascinating. This chart completely flips the old story on its head. For decades, we all thought of this as a hospital-acquired bug, right? A nosocomial problem. But this big Australian study found that over half of all bloodstream infections were actually community-associated. That is a major paradigm shift in how we should be thinking about this organism.
All right, this brings us to the bench. How do we actually spot this thing in the lab? It's a great case where your classic old-school microbiology skills are every bit as important as our fancy automated systems.
So under the microscope, it's your standard gram-negative rod. Nothing too exciting there. But on the plate, oh, it's a showstopper. You're looking for these dry, wrinkled, leathery colonies that are really tough and literally stick to the agar. That morphology is your big red flag. But, and this is a big but, be careful. On subculture, they can sometimes lose that wrinkly look and go smooth. So always trust your primary plate.
Biochemically, it's pretty active. It's oxidase positive, catalase positive, and it's a potent denitrifier. But the absolute key differentiator from its more famous cousin, Pseudomonas aeruginosa, is the arginine dihydrolase test. Stutzerimonas stutzeri is negative, Pseudomonas aeruginosa is positive. It's a simple but absolutely crucial test to tell them apart.
Now, for a definitive ID, your modern methods have some pros and cons. 16S sequencing, it isn't going to cut it here. Not enough resolution. Sequencing the RPOD gene, that's the recommended way to go for an accurate ID. And what about MALDI-TOF? Well, it's fast for sure, but its success completely 100% hinges on having an updated database that actually recognizes the new Stutzerimonas name. All right, so you've nailed the ID. Now comes the really hard part. How do you treat it? This is where we, as a community of labs and clinicians, hit a major roadblock. We are operating in a total guideline vacuum. And this is the million-dollar question, isn't it? EUCAST has no specific breakpoints for this bug. The Dutch SWAB guidelines don't have formal protocols either. So what are we supposed to do? How can we confidently report susceptibilities and help guide treatment? Do we just use Pseudomonas aeruginosa breakpoints? Or maybe the breakpoints for other non-Enterobacterialis? It's a genuine clinical dilemma we face every day. And this problem gets even tougher in pediatrics. The data is just incredibly thin. We're talking about a handful of case reports. Treatment has to be highly individualized, and a lot of the time, the dosing is just borrowed from what we do for Pseudomonas aeruginosa, which, let's be honest, is far from ideal.
But here is the good news. There is a light at the end of this tunnel. A brand new proposal from the Clinical and Laboratory Standards Institute, CLSI, is finally giving us the evidence-based path forward that we've all been desperate for.
So listen up, because this table is everything. This is from the upcoming CLSI M45 guideline for what they're calling POPA, Pseudomonas other than P. aeruginosa. And that group includes our bug, Stutzerimonas stutzeri. These tentative breakpoints are the most current, data-driven tool we have. They finally give us a real framework to interpret MICs and help guide effective therapy, moving us away from all the guesswork. This is a game changer.
Okay, so now that we have a much better handle on how to identify it and how to treat it, let's talk about prevention. How do we stop these infections from even starting? The absolute key is to remember its environmental nature.
And this case study just illustrates the point perfectly. There was an outbreak of wound infections, but the source wasn't another patient. It wasn't a healthcare worker. It was a communal pot of saline-soaked cotton balls. The fix was simple. Stop using the communal pot. For an environmental bug like this one, the critical infection control question is never who, it's always where.
So let's wrap this all up with the key things to remember. First, it's Stutzerimonas stutzeri now. Second, it's mostly a community pathogen, which is a huge shift. In the lab, you've got to blend those classic observation skills with modern tools. For susceptibilities, those proposed CLSI M45 breakpoints are your best friend. And for prevention, think environmental sources, especially water.
You know, this quote really nails it. The whole story of Stutzerimonas stutzeri is just a perfect reminder of how dynamic our field is, and that our work is truly never, ever done. There's always more to learn.
And now I really want to hear from you. Have you been seeing this organism in your lab? What kind of challenges have you had with ID or reporting susceptibilities? Please share your experiences down in the comments below. Let's get a conversation going.
Seriously, your insights are what make this community so valuable. And if you found this explainer helpful, please hit that subscribe button for more deep dives into the bugs that really matter in our labs. Thanks so much for joining me. I hope you found this useful.